Hello everyone, today in this lecture we will explain the gram-negative root bacteria, the large family, which is called Enterobacteriaceae. And this family are commonly found in the intestinal tract of the human and animal, and many bacteria in this family are part of the normal microbiota but also can cause a serious infection. Today in this lab we will understand the general characteristic of this family of the bacteria, classification, how diagnose this bacteria in a lab, and the disease that are caused by this large group of gram-negative root bacteria. The family of the Enterobacteriaceae, the most common gram-negative root bacteria. They naturally habitat the intestinal tract of the human and animal, therefore it's called the enteric bacteria. It also known as the coliform bacteria. Why this group or this family of the bacteria is clinically important? Because this family, the gram-negative Enterobacteriaceae, along with staphylococci and streptococci, among the most common bacteria that are cause the disease in humans, such as UTI infection, gastrointestinal infection, septicemia, and others. Now, the general characteristic of the Enterobacteriaceae, this group of bacteria are short gram-negative root bacteria. It means it appears under the microscope as a pink bacilli when we do the gram stain. Why? Because it's gram-negative, it appears as a pink or red color under microscope when we do the gram stain. And that can grow rapidly on a standard laboratory media such as blood agar, nutrient agar, and this group of the bacteria is catalyst positive. It means they can break down the hydrogen peroxide into the water and oxygen. And it's oxidase negative, and that's important. When we do the oxidase test, this group of bacteria, they are oxidase negative. That means they don't have the oxidase enzyme. And we can identify by the ability to ferment the lactose on maconge, such as E. coli. They have the ability to ferment the lactose on maconge agar and produce the pink colony. Or other, we can identify also by other biochemical reaction. Another important characteristic, they can reduce nitrate to nitrite, and they are generally motile with a preterigus flagella. However, some species are non-motile, such as Shigella and Klebsiella, and they are facultative anaerobic. That means they can grow with and without the oxygen, and can ferment wide range of the carbohydrates such as lactose, glucose, and have a complex antigenetic structure. That means they have several types of the antigen on their surface, and this is important for classification and identification, and have a variety of the toxin and virulence factors that help them to cause the disease. And the Enterobacteriaceae family contain many important genera, such as Escherichia, Shigella, Salmonella, Enterobacter, Klebsiella, Serratia, Proteus, and others. Some species are normal flora, but can become opportunistic and cause the disease. For example, E. coli is normally present in the intestinal, but may also cause UTI infection, diarrhea, and others. However, some species are always pathogenic, such as Salmonella and Shigella. It considered pathogenic to humans. As I mentioned, all members of this family are gram-negative root bacilli. It appears under the microscope as a pink root short bacilli, sometimes arranged single or in a small group. And example, you can show in this slide the Escherichia coli. It appears as a pink root bacilli under the microscope. Now, the natural habitat, what it means. As I mentioned, most Enterobacteriaceae are commensal in the human intestine. It normally present as a normal flora in the human intestine. For example, E. coli, Klebsiella, Proteus, Citrobacter. However, some species are not normal flora and are always pathogenic, like Salmonella and Shigella. It considered pathogenic to human. And here in this diagram, you can show the human body and the infections caused by the Enterobacteriaceae. These bacteria can cause infection in many body systems. It can cause the UTI infection, diarrhea, pneumonia, sepsis, meningitis. For example, Klebsiella pneumoniae, it can cause pneumonia. And E. coli is the most common cause of the UTI infection. Classification of the Enterobacteriaceae. We can classify the Enterobacteriaceae by doing the classical method, which is commonly used in microbiology lab. It depends on the ability of the bacteria to ferment the lactose, and mainly in the maconge agar. And according to the modern taxonomy or modern classification, it not only depends on a biochemical properties like the carbohydrate fermentation, but also on the common morphological like the shape, gram reaction, along with a DNA-based composition and the genetic similarity. For example, Enterobacter and Klebsiella, both of them is a lactose fermenter. By doing the genetic study, we can differentiate between them because the DNA is different. This method is more accurate. However, in most clinical labs, the lactose fermenter method is still used as a first step for identification because it's a quick method. As I mentioned, the most common method or classical method to classify the Enterobacteriaceae is according on a lactose fermentation and mainly on Macongi agar. We can classify into three groups. The first group, the lactose fermenter bacteria, they ferment the lactose on Macongi agar and produce the acidity, and the colony of this group of bacteria appear as a pink colony due to the fermentation of the lactose and change the color of the indicator in the media, like Escherichia coli and Klebsiella. The second group is a non-lactose fermenter bacteria. They don't ferment the lactose, and the colony of this group of bacteria appear as a colorless, like Salmonella, Shigella, Proteus. The third group is a late lactose fermenter, like Shigella soni. They ferment the lactose, but the fermentation is slowly. It requires more time to ferment the lactose. For example, after 2 days may produce the pink color colony. Required more incubation period because it's a slowly lactose fermenter bacteria. And there are several selective and differential media used for isolation of the Enterobacteriaceae. The most important one is Macongi agar. It's a differential media. It can differentiate between the lactose fermenter from the non-lactose fermenter. As you can see in this image, the lactose fermenter bacteria produce the pink colony on Macongi agar, and the non-lactose fermenter produce the colorless colony. And the second media is eosinamethylene blue agar, and it's mainly used for identification of the E. coli. As I explained in a previous lab, E. coli produce a metallic dark green shine on this type of media. And Salmonella Shigella agar is mainly used for Salmonella and Shigella. Trouble sugar iron used to detect the sugar fermentation, gas production, and hydrogen sulfide production. How can diagnose this large group of the bacteria in a lab? The first step is a specimen collection. We collect the specimen according to the site of the infection or the infection type. In the case of the UTI infection, we collect urine. If they are bacteremia, we collect the blood. If they are pneumonia, collect sputum, and so on. So it depends on the site of the infection and the infection type, we collect the specimen. The second step is a microscopic examination by doing the smear and gram stain. As I mentioned, this group of the bacteria is gram-negative root bacteria. It means under the microscope appear as a pink root bacilli bacteria. If we do the capsule stain, the large capsule may indicate the Klebsiella. The specimens are cultured on blood agar and Macongi agar. The differential agar for this group of bacteria is Macongi agar. For quick identification of the gram-negative enteric bacteria, we should culture on a Macongi agar is a differential media for this group of bacteria. Now, the Enterobacteriaceae may have several virulence and antigenetic factors that help them to cause the disease and help this bacteria to colonize, adhere to the host cell, produce a toxin, as some of them may have the plasmid that contain the gene for antibiotic resistance. For identification and classification, many Enterobacteriaceae have important antigen. The O antigen is a somatic antigen. It means it's a part of the bacteria structure. It's located in a cell wall of the bacteria. It's a heat stable. It's not destroyed by the heat. The H antigen is located in the flagella and is a heat labile. It means destroyed or damaged by the heat. The H antigen is located in the flagella. It means the non-motile bacteria like Shigella and Klebsiella, they don't have the H antigen because they don't have a flagella. As I mentioned, the H antigen is located in the flagella. The K antigen located in the capsule surround the bacteria cell, and it also heat labile. And this antigen is important for serological identification of the Enterobacteriaceae. Now, the members and disease caused by the Enterobacteriaceae. Members of the Enterobacteriaceae are commonly found as part of a normal flora of the intestinal tract, may also exist in a small number in the upper respiratory tract and genital tract. And in their normal location, these bacteria usually do not cause the disease and may even help in a normal intestinal function and nutrition. However, they become pathogenic when they spread to other parts of the body outside the intestine. And the most common infection occur in the urinary tract, biliary tract, and abdominal cavity. They may also affect other sites such as bloodstream, lung, prostatic gland, bone, or meninges. And some species, such as Serratia marcescens, act as opportunistic pathogens. That means they are mainly cause infection when the host immune defenses are weakened, such as in infant, elderly individual, or immunocompromised patient. And in severe cases, they may enter the bloodstream and cause sepsis. Now, after understanding the general pathogenesis of the Enterobacteriaceae, we will focus on one important member of this family, which is Enterobacter. Enterobacter is considered as opportunistic pathogen, especially in hospital setting. And this bacteria have several important characteristics. We can identify in a lab as it can ferment the lactose. It means they produce a pink colony on Makongi agar and have a capsule that gives the mucoid appearance, and it's motile. That means they have the flagella, use the flagella for movement. And Enterobacter are commonly associated with hospital-acquired infection, nosocomial infection, especially in patients with a weak immune system that are used the medical device. Example of the infection, pneumonia, especially in a ventilated patient, urinary tract infection, common in a catheterized patient, and wound infection after surgery, and device-associated infections such as the catheter and IV lines. Now we will move to another important opportunistic pathogen within Enterobacteriaceae family. Serratia marcescens is commonly associated with a hospital-acquired infection. It is the most common opportunistic pathogen. That means they cause the disease in patients with a weak immune system, especially those who are hospitalized. It can cause several serious infections such as pneumonia in a ventilated patient, bacteremia, bloodstream infection, endocarditis, and those infections are common in patients who are using invasive medical devices such as intravenous line, urinary catheter, and ventilator. For example, a patient in ICU with a central venous catheter may develop bacteremia caused by Serratia marcescens. Now, in a lab, they have the most important characteristic. Most strains are non-pigmented, but about 10% produce the red pigment, and that is help in identification in a lab. And it is DNA positive. We can do the DNA test to differentiate the Serratia marcescens by other Enterobacter, as Serratia marcescens is DNA positive. And regarding the antibiotic, is antibiotic resistant. Is often resistant to the penicillin, and the treatment usually required the more strong antibiotics such as the third generation cephalosporin. Another member of the Enterobacteriaceae, Proteus species, are part of a normal intestinal flora, but can cause infection only when they leave the intestine and enter other body site, and is commonly associated with a urinary tract infection, UTI, but can also cause the pneumonia, bacteremia, and other infections, especially in hospitalized or catheterized patients. And that's make them important nosocomial pathogen. Now, the high motility help them to spread easily and evade the urinary tract. And in a lab, they show the characteristic of the swarming growth on agar. That's help for identification. And the key feature is production of the enzyme urease that break down the urea to ammonia, make the urine alkaline. And this alkaline environment help to formation of the urinary stone. For example, patient with a recurrent UTI caused by the Proteus may develop kidney stone due to this effect. And thank you for your attention, and that's all about the part 1 of the Enterobacteriaceae.